Have you ever asked yourself the question of how complex biological systems developed from cells to humans and how they work? Many characteristics of living systems derive from mutual interactions of biomolecules. Interactions between two basic types of biomolecules, nucleic acids and proteins, lay the base for biological processes. You're seeing one of the protein-DNA complexes right now, a complex of DNA in the nucleosome core. In our laboratory, we study both proteins and DNA. We solve their structures, measure their interactions, stabilities, and dynamics in space and time. We try to elucidate behavior of biomolecules in connection to their biological functions. Our results are used in diagnostic, medical, and biotechnological fields of research and development. The possibility of biomolecular structure determination, which became available in the second half of the 20th century, opened up a new view on biology and helped us understand many key processes in life. Nucleic acids and proteins display fascinating architecture wherein molecule function is encoded. But the path to solving a molecular structure is lengthy. The process starts in the laboratory, where cell cultures are grown and thereafter provide studied proteins. Most of our experiments require an active and highly pure protein. It's obtained from a cell suspension by way of purification that uses high-performance liquid chromatography methods. As the next step, pure proteins are characterized. We measure, for example, their thermal stability, stability in different types of solutions, or a degree of molecular ordering. Throughout the process, we work with tiny volumes of protein solutions under clearly defined conditions and in a sterile environment. For all molecular analyses, we utilize our involvement in the European infrastructure Instruct-ERIC. One of the most complex methods we use is crystallography, which determines an accurate arrangement of atoms in biomolecular structures. The first step in determination of a molecular structure is crystallization. In case of biomolecules, it is a complicated and time-consuming process which takes weeks or even months. Although people carry out the majority of the work, specialized robots can perform some activities such as liquid preparation into crystallization plates or monitoring of progress of crystallization in the crystallization hotel. We obtain information about the molecular architecture from crystals using scattering of shortwave radiation, so-called diffraction, and by consequent mathematical evaluation of measured data. Recently, we've solved several protein structures. Specifically, we investigate the structure and function of interleukins and interferons. They are proteins especially important in the immune system. Their dysregulation causes serious autoimmune diseases such as psoriasis, rheumatism, and multiple sclerosis. Therefore, in addition to the structural characterization of interleukins and interferons, we strive to modify their properties. We search for protein variants that could be used in medical treatment or diagnosis. Bioinformatics is also a significant part of the research of proteins and nucleic acids. It analyzes large collections of data and examines their biological importance. In our laboratory, we study structural variability of nucleic acids and their interactions with proteins. Our website, dnatco.org, offers a unique classification of nucleic acid structures. On a long-term basis, we study the key biophysical phenomenon of hydration. We're able to execute our bioinformatic projects thanks to a participation in the European Bioinformatics infrastructure, Elixir. And last but not least, we study dynamic, time-variable aspects of interactions between proteins and DNA. In our laboratory, we devote great attention to this research. Thanks to our collaboration with the Institute of Physics of the CAS, the ELI Beamlines Laser Facility, we can study the dynamics and interactions of potentially technologically interesting transcription factors, meaning proteins controlling the synthesis of other proteins in cells.